So, thank you for the invitation to be here joining this focus on Latin America and the Caribbean roundtable discussion. I will be showing you today some of the results of the AmaZOOmics project initiative, which started in 2021 and seeks to bring together the Vale Technology Institute, ITV, several Brazilian and international universities and organizations, and the Vale's Biopark to generate genomic resources intended for the conservation of Amazonian biodiversity. ITV is a private research institute established in 2010 and devoted to sustainability science, including a large number of research groups ranging from geology, environmental science to genomics. Here are some photos. You can see here some photos of ITV situated in Belém, near the mouth of the Amazon in Brazil. And you can also see some pictures of the sequencing machines that we have there, including on the top there, Sequel 2 PacBio and 2 Illumina machines. Vale's Biopark, you can see on the inset here, is situated in the so-called Carajás Conservation Mosaic, a continuous area that includes several conservation units in southeastern Amazonia. Vale's Biopark houses a zoo that's open to the public and which hosts animals found hurt or confiscated from the illegal trade business or from illegal pet owners. Most of these animals have known provenance and constitute great resources for obtaining high-quality DNA extractions for high-Fi and high-C genome sequencing. Vale's Biopark has a dedicated team of vets who can obtain non-invasive genetic samples, mainly blood, from several Amazonian vertebrate species, including many that are currently endangered. The Carajás Conservation Mosaic includes several hundreds of thousands of hectares, and its vertebrate fauna has been and will continue to be sampled in the context of the AmaZOOmics project, mainly for vertebrates at this point. We have been actively collaborating with the VGP, with phase 1 of the VGP project. And so far, one reference genome has been produced under this partnership, and that's the Harp Eagle genome, which has been already assembled and is being currently annotated and should be sent out for publication this year still. These are the other phase 1 species that that ITV will be contributing with. You're talking about all these. We have already collected genetic samples from Vale's Biopark this year for all these species, including monkeys, jaguar, hyacinth macaw, and several other species. We have been collecting samples in liquid nitrogen. They are flash frozen immediately upon collection. So we are following gold standard procedures concerning the collection of genetic material. This is a picture of Geronimo, the AmaZOOmics postdoc, collecting samples at Biopark. We will still carry out a second collecting expedition, so to speak, to Biopark to sample these additional species there: tapir, howler monkey, peccaries, caiman, and turtles. And these samples will be also sent to the VGP headquarters in New York for high-Fi and high-C sequencing. Um, I have shown you all those species before that will be sent to New York to be sequenced at the VGP headquarters, but starting this year, we'll be already producing chromosome level reference genomes in-house. So at ITV, using our sequencing machine that I just showed you. And amongst these genomes, there will be genomes for the ant birds, for endemic ant birds, as well as for bellbird that is regarded as an endangered species, and local fish and turtles, including one of them is the arapaima or paiti or piracu fish, one of the largest freshwater fish in the world, and a very important component of the sustainable fishery industry in the Amazon. We are also resequencing, I should say, hundreds of individuals for their whole genomes for species of interest, including the harpy eagle again. We have already a landscape genomics project going with preliminary results. And here you can see a plot showing the population structure found in the harpy eagle. You can see different points and clouds of points denoting populations from different biomes in Brazil. And the same thing is true for the hyacinth macaw. We are also sequencing dozens of individuals from its range and assessing the degree of population structure and this species based on whole genomes as well. This work is being done with the support from 2 PhD students and 1 postdoc as well. We have also begun to explore the adaptive genomic potential across the moisture gradient in Amazonia. And that has some important implications for designing species resilience strategy, nature-based solutions for the conservation of the Amazonian avifauna in face of global change. Our first results, these are whole genome sequenced for several populations of a non-migratory endemic passerine. And these are PSMC plots showing that how demographic trends were like between the last interglacial and last maximum glacial. And you can see that the effective population size in southeastern Amazonia, in the southeasternmost tip of the Amazonia, the effective population size there are much smaller than in other parts of the Amazon. So these are really, these patterns reflect probably that these areas are less climatically stable, and concerning the long-term persistency of humid forest, and possibly these reductions in population and sizes were also accompanied by signatures, selective sweeps maybe, of adaptation to these local and more seasonal and drier climates that may have a secret to tell us about the resilience in these populations. Thank you. That's pretty much a summary of what we have done so far. Thank you very much. And feel free to get in touch with me to ask any questions, and also to partner up with us at ITV. Scale up is in the horizon, and we have the ambition of reaching 350 plus genomes by the end of 2027. And this is a challenge in itself. So we are reaching out for collaborations across the board to make this happen. Thank you.